Enzyme Inhibition by Sumandeep Kaur
and Jigma.
Our presentation is based on a recent publication about the inhibition effects of furfural on alcohol dehydrogenase, aldehyde dehydrogenase, and pyruvate dehydrogenase. We will be using these two figures to show the effect of the inhibitor furfural on pyruvate and pyruvate dehydrogenase. The aims of this presentation are to explain what enzyme inhibition is, the types of enzyme inhibitors there are, how to distinguish between the different types of inhibitors, what pyruvate and pyruvate dehydrogenase are, and the effect of furfural on them. What is enzyme inhibition? Enzyme inhibition is when the activity of an enzyme is inhibited by the binding of a specific molecule, decreasing the rate of reaction. From the image, we can see that the specific molecules are inhibitors which bind either to the active site or on a different site on the enzyme and prevent it to work in its normal manner. There are two types of inhibitors, reversible and irreversible. Irreversible inhibitors inactivate the enzyme either by binding to it covalently or non-covalently. Reversible inhibitors are in contrast with irreversible inhibitors, as they bind to the enzyme through weak interactions such as hydrogen or ionic bonds. There are three types of reversible inhibitors: competitive, non-competitive, and uncompetitive. Competitive inhibitor resembles the chemical structure of the substrate and competes for the same active site as the substrate. If substrate concentration is increased, it can displace the inhibitor. Non-competitive inhibitor binds at a different site from the substrate and modifies the substrate's active site, therefore the substrate can no longer bind to the enzyme. Increasing the substrate concentration would not overcome the non-competitive inhibition. Uncompetitive inhibitor binds to the enzyme-substrate complex, forming the enzyme-substrate inhibitor complex, and prevents it to form the product. Uncompetitive inhibitor cannot be overcome by increasing the substrate concentration.
In order to identify which inhibitor is involved in the reaction, enzyme assay can be carried out, and from the data collected, Michaelis-Menten plot can be plotted, but from this plot it is very hard to determine Vmax and Km. Easy and more accurate way to determine Vmax and Km is by using Lineweaver-Burk plot, as Vmax and Km are the intercept on the plot. Michaelis-Menten equation can be converted to a double reciprocal plot, which is a more convenient representation of the data. In competitive inhibition, the maximum velocity of the reaction is unchanged, while the apparent affinity of the substrate to the binding site is decreased. Any given competitive inhibitor concentration can be overcome by increasing the substrate concentration, in which case the substrate will outcompete the inhibitor in binding to the enzyme. When non-competitive inhibitors are added to a reaction, Vmax reduces as the enzyme can no longer function as well as it could in the absence of the inhibitor. Note that the Km for the reaction does not change, because while the enzyme isn't catalyzing the reaction, it can still bind to the substrate just as well as it did before. And remember, Km is the measure of the affinity of the substrate to the enzyme. Uncompetitive inhibition decreases the maximum velocity as well as the Km. Both Vmax and Km are reduced by equal amounts. Increasing substrate or enzyme concentration will not overcome the concentration of uncompetitive inhibitor.
Pyruvate and pyruvate dehydrogenase. Pyruvate is the end product of glycolysis, which is converted into acetyl-CoA that enters the Krebs cycle. Pyruvate dehydrogenase is an enzyme which catalyzes the oxidative decarboxylation of pyruvate into acetyl-CoA, which is then used in the citric cycle for cellular respiration. The equation below shows the reaction of pyruvate to acetyl-CoA. The effect of furfural on pyruvate and pyruvate dehydrogenase. The publication's conclusion suggested that the presence of an inhibitor of furfural can stop both pathways leading to acetyl-CoA from pyruvate via pyruvate dehydrogenase. This can be seen in figure 1a, where the pyruvate dehydrogenase's relative activity percentage is the lowest even at a low concentration of the inhibitor. This suggests that the pyruvate dehydrogenase is being inhibited by furfural. As the concentration of furfural is increased, the activity percentage of pyruvate dehydrogenase decreases even further until it reaches 0. Figure 4b from the publication also supports the conclusion, as it shows that increasing the pyruvate concentration does not overcome the inhibition activity of furfural. This also shows that furfural had the effect of a non-competitive inhibitor on pyruvate dehydrogenase. These are the references used for this presentation. Thank you for listening to our presentation.